Respected faculties, good evening to all. I welcome all of you for the day 4 session AICTE-ATAL sponsored FDP on intellectual property rights. So today, as we have two sessions, session 1 will be handled by Dr. Karthik. Dr. Karthik has an extensive expertise in actinobacteria research, phage therapy, synthetic biology, and drug discovery strategies. He's a microbiologist and founder of Arc Gene and Sasi Nutraceuticals Vellore. He did his PhD degree in microbiology from VIT University and postdoctoral research at Shanghai Jiaotong University, China, under the guidance of Professor Xiong Li, and then second postdoctoral research fellowship at again ECU ST Shanghai, China, under the guidance of eminent scientist Professor Lixin Zhang. He is placed among top 2% of all scientists in the world, which is published by Stanford University report by the Elsewhere as reported in the October 2023. He has developed CRISPR-Cas9 based diagnosis kit and a new cloning method which is patented in the country China. He has published around 91 research papers which includes Nature Communications, Nucleic Acid Research, RSC Chemical Communications, Nanoscale Advances, Nanomedicine, PLOS1, and Parasitology Research. Dr. Karthik's H index is about 29 in Scopus, and he has published 5 books and holds 6 patents. He also developed 6 products, and all the 6 has been commercialized now. And presently, he is a communication committee member of International Chemical Biology Society USA and president of Society of Chemical and Synthetic Biology India. He also got prestigious Young Scientist Award from various associations, which includes Indian Association of Applied Microbiologists, Outstanding Research, and also Best Scholar from VIT University. He is also serving as Associate Editors of Frontiers in Microbiology and editorial board member of Discover Applied Sciences, which is published by Springer. And also, he has got several research grants from DST, DBT, DST Ignition Grant, and DST Nidhi Prayas for his startup. Once again, I welcome you, sir. The session is yours.
Yeah, thank you very much, Dr. Shakti. So first of all, I thanks to the management of PSG and Dr. Shakti and the entire team for your kind invitation and introduction. So it's honor to me to interact with all the faculties here. I hope here different field of people is there. So the how the AI can be implemented in biological field. So I'll give it very basic introductions to how all the fields can be applied, and future if any people need any help for product development also, our company is working not only AI in biotech, we are also working with the AI in other fields like ECE, biosensors, with okay, all the fields we are currently working, and for that we have grants also. Okay, so it's not only just a presentation. I want to make a like an interaction section. So after the short presentation, I want to interact one by one how the AI can be used in a field. Any doubt we can ask. Okay, so now I will go to that share my things. So can I start to share my PPT, Doctor?
Yes, sir. You can start presenting your slides.
Yeah, just a minute. So now my PPT is visible, doctor?
Yes, your slide is visible, sir.
Yeah. Now I'm starting. So if anywhere, if PPT is not moving, please inform me, sir.
Yeah, definitely.
Yeah. So good evening to one and all. So it's really, as I told, we are working several fields of AI-driven drug development, drug discovery, as well as health sectors. So first, I don't want to explain much for you people what is artificial intelligence nowadays, from basic chat GPT to DeepSeek, everyone working for either taking notes and others. This is a way it's going on. But we need much beyond that. Okay, so what are the things happening in the world wide means AI-based drug delivery and drug discovery. Okay, and several app development recent days is going very high level. Even ADMT prediction, clinical trial design, in all the fields is going very fast. So as I told in drug delivery, particularly smart drug delivery system, AI-driven design for nano carrier, modeling drug release, digital twin models in trial, and rapid screening and accurate protein folding predictions. As an adaptation level, now we are in 2025. In within another 3 months, we are in 2026. But as per 2023, you can see that academia, how much percentage of AI is happening in industry, how much, particularly AI-based biotech startups. This is a very important thing. You can see that how extensively they are using. 20% startup. If you go to IIT Chennai, VIT, SRM, private lot of reputed institute, if you go, the AI-based companies are growing very fast, as well as the funding. Main thing is the funding. As a faculties, if people like to write the fundings, okay, particularly who are all written in bio AI, particularly last month, BIRAC given the call for bio AI project. Okay, they are ready to give unlimited funds. There is a no limitation. Okay, so those have interested, so faculties can interact with the concerned AI company who is related to your field, and you can submit the BIRAC proposal. Particularly, they like to institute and industry related projects only. They're asking collaborative projects. So it's a day by day is increasing in every field. What you should understand. So as I said, that current state in AI drug delivery is estimated that around 30% of new drugs are from AI discovered. Then your mind will think, sir, what then no drugs are discovered in the previous year. I will show some data, how much low it is, you will understand. Okay, so first of all, you should know why the drug discovery is needed. See, if anyone ask what is a dangerous disease, if any non-subjective people also, everyone will tell us cancer or HIV. These are things we will tell, right? But the real story is the most dangerous, we use the term called antimicrobial drug resistance. That means very simple term. If I want to tell, very small disease also, the previous drug is no more working. Like we all know that typhoid, everyone in life somewhat we cause the disease called typhoid, right? So typhoid is caused by Salmonella typhi, basically. Biotech people aware of that, but the drug is what is existing is no more working, because when over usage of this drug keep on to this bacteria, it get resistant. Okay, due to that, it's no more working. Like this cholera, tuberculosis, all the major diseases, the existing drug is no more working. If this case continuously go, the WHO predicted that 2015, 10 million people will die due to the aim, or not due to cancer or diabetes, not other diseases. So it's a very serious threat. So what is how we can solve this issue? So one of the natural, the olden time we use what kind of treatment? We all know that herbals is one of the key features it have. New antibiotic producing capability have. So all the antibiotics isolated from that, but we're not aware of that. After we come to the modern era, allopathy medicines come, we dropped totally the old medicines and we enter into that this modern era after the penicillin discovery. You can see this red line and blue color line. The blue color line is indicate every time the new drugs coming, but the line clearly indicate that the novel drug we called as a lead structure, like a penicillin kind of ring, discovery is getting low day by day, because to find one drug it take around 20 years, including after licensing everything, and they want to spend millions of dollars. After 5 6 years only they come to know this drug is no more working, we should drop this project. That is the biggest problem in drug discovery. But what is the opportunity with us? After the new whole genome sequencing era came, this cost effective is reduced. Okay, for that, every researchers to find the new drug they are working after medicinal plant, one of the important bacteria called actinobacteria. Okay, it's a fungi-like bacteria. I don't want to go very deep on this. So very simple term for everyone understanding. We all like rain smell, right? The rain smell due to this particular bacteria. It produces a compound called geosmin. When it react with water, it will produce a pleasant smell. Okay, so it's like our day-to-day life we are living with this bacteria. Okay, it's high GC content bacteria. The interesting thing about for all, nearly 42% of antibiotic what we are using in the market, all are isolated from this particular bacteria only. After medicinal plant, this is a huge source for us. Okay, this is the first picture. It's naturally present in the soil. The next you can see some beautiful picture of this bacteria. We call it as in biotech term is a bacteria like fungi because you can see this like a mycelium like appearance. In 2015, three people shared the Nobel Prize. One is Sandoshi Umra to find the drug called ivermectin. I don't know how many people are aware that during COVID time is one of the drug of choice in starting days. Okay, this is a person who really discovered. Then William Campbell, along with him, shared the Nobel Prize. He is a person who find that the ivermectin can be used for night blind diseases. For that, he could share the Nobel Prize with him. And Professor Yu from China, who find the drug or malaria, artemisinin from plant sources. Okay, this three people share for natural product drug discovery. They share the Nobel Prize in 2015. As I told now, we come to know this is a bacteria. Okay, then how to find that this is a drug molecule, it have the new one or not. So generally, as I told, the olden time they will do traditional agar well diffusions, those very old method they will use. But now after the sequencer revolution, so this is a starting time sequence machine. You can see the size of this. The price is around 1 crore to 5 crores. It's not easy to handle. It need more expertise in biotechnology people and even computational people, particularly computational programming people needed for this. It's a combination of two people. That's why the bioinformatics course introduced, but still lot of programmer are lagging in this field. Okay, for to overcome this issue, only Oxford University recently discovered the technology called Minion. It's called as a nanopore. You can see this now, very handmade. Just you can carry anywhere with one laptop on this. There's no need any expertise to run this machine. Just to have a look this video how this machine is working. This is a combination of ECE and biotechnology. You saw this video. It once the DNA enter into the pore, it converted into the DNA converted into the electric signal. Okay, here the electronics play a role. Then the electric signal converted into the biological data. Okay, so due to this new revolution of this technology, the human genome price cost also reduced a lot. So the future, we no need to do any X-ray, ECG kind of things for cancer kind of diseases. Just they will tell, go and do this particular marker, do the sequencing, because the sequence will complete within 30 minutes to 24 hours. Depends on the target size. In the short span of time, it will be completed. So due to that, the prediction hereafter, we carry all our whole genome sequence in CD. Okay, then some AI tools will be there. It will help to predict. Even no need any doctor assistant. Doctor will play the secondary role only. The first, the prediction will do everything AI. The secondary will uh, data prediction, and the treatment only will be provided by the doctors. Okay, such a revolution is happening. The combination of such high technology with AI. Okay, so what we done in this? So the worldwide, as I told, the actinobacteria, 10,000 actinobacteria, whole genome sequencing we done first time in the worldwide. Then we found that a lot of interesting molecule present in this for cancer, malaria, flu, diabetes, even AMR, everything. Not only for actinobacteria, we also do done for fungi too. After doing sequence, in biology, people know that we submit this all the data in NCBI website. Currently, 60% of actinobacteria what we submitted in our lab itself. Okay, but what is the real challenge in this? Most of the genes we term use the call silence. It's not expressing in the bacteria. Okay, due to that, we are unable to predict this what kind of antibiotic it will be produced. Those information is not available. How to activate, what kind of protein or what kind of drug it will be produced. Everything is the biggest challenge for us. So for that only already that some AI-based tools are they developed. It's called antibiotic secondary metabolite shell. Okay, the first before this, after I told after sequence done, the assembly is the biggest challenge for biotech people. As I told, for this each lab needed one biotech as well as one computer science people. Okay, even commonly bioinformatics people is we need much people, but as I said, most of the bioinformatics people not aware of how to do the programming because this all the assembly softwares are Linux programming. Who are good at R or Linux only they can do. Okay, to overcome this issue, currently our company made that map my genome like that. One AI based softwares we made. Currently it's under patent. Okay, so I'm not a disclosure much on this. So any even non-computing people who don't know even AI, just they can submit the raw data which is generated from the nanopore. You saw that, right? The electric signal. The electric signal once it is submitted to the software, it automatically converted in the nucleic acid sequence. Okay, so it no need any expertise or you don't need any Linux programming skill. This AI tool will help you completely to assemble properly. Assembly is nothing but the which is head tile like that. In human we have starting should be head, below should be that leg, right? So like this foot, this is the order we needed to assemble in bacteria and fungi also. This is the biggest challenge. It will take months wise to solve this issue, but what the AI tool we developed, it will complete for each bacteria in 30 minutes. This AI tool will be complete within 30 minutes just to see this revolution in AI. So due to the scientist can be go through immediately that what kind of drugs are present in this bacteria and how to express those genes. So once the assembly happened through the AI tool, then we will go to the another AI tool called, as I told, antibiotic and secondary metabolite analysis shell. In this, if you submit your whole genome, it will clearly explain that what are the antibiotics are exist in this, what are the drugs are new in this. You can see this for a small example here. First, the cluster one indole, most similar known cluster is showing dash. That means what? There is no existing molecule present in this. Okay, the next drug it showed it matched with the previous drug 17%. The next drug it showed it's a 50% match. The fourth one it shown 100% match. So in bioinformatics, we the cutoff values will keep it okay. So more than 30% we consider as it's a known molecule. Less than 30 we'll consider as a new molecule. Okay. In this concern, if we see, as I told, 10,000 actinobacteria genome sequence be done. When we analyze everything, you can see 16, 17, and if you see some genome, completely no. New drugs are there. You can see everything dash, dash, dash. That means what? There is a no existing molecule matched with this. And all these clusters can able to produce new drug. As I told that olden time, it will take several years to come to this stage. But after the revolution of this AI and others, now it happening very fast. That's why in the second slide I told now 30% new drugs are discovered by AI based drug discovery methods. Now you can understand why the AI need and what it's a play major role in drug discovery. Okay, after going this, as I told, the silence gene can be expressed. Okay, so to find which gene should activate, for that we need once again AI driven tool. Okay, what is the exact promoter or what kind of gene should be activated? Okay, so in the normal method, you're unable to activate, we go to the genetic manipulation, then we'll proceed further. So the first genetic manipulation method called as the heterologous expression. Once the particular whole genome, as I told, with the AI tool, we assemble it. Then we'll analyze by another AI tool what are the genes are responsible for this. That we clone into the another system. Then we'll express into that. This is called heterologous expression. Okay. Now after this revolution, this technology can be complete within 2 weeks to 4 weeks for any bacterial or yeast strains. But once again, every methodology has some own drawbacks, right? So for that only the next new technology called CRISPR. The CRISPR is nothing but cluster regular interface short palindrome repeats. It's nothing but for all the department people to understand easy way. This is the immune system of the bacteria. Everyone know the human we have immune system, right? The scientists recently found that the bacteria which is causing infection to us that also have the immune system. So for what it need the immune system? Okay, so we all studied maybe the school books, even who not in biology on everyone in 10th school book itself, we will read like phage, bacteriophage or phage, we will use the term, right? The phage is the biggest enemy for bacteria. It will go and sit and it will bite the bacteria and it will die. This is a story we read so far, right? You can see this image, everyone, this phage. This is a phage. Once it sits on the bacteria, it releases DNA. Then multiple phage virus will be, phage is nothing but virus, grow inside much and it will blast and thousands of new virus will be released. But this story is no more. So what happening now when it releases this DNA, the bacteria considers some enemy enter inside, then it produce some kind of antibodies, kind of things called only nuclease enzyme we use in biological term. Okay, it cut the DNA and the bacteria will escape. The scientists found this thing only. For that only the given name is CRISPR. For this identification, Jennifer Doudna and Emmanuel Charpenter share the Nobel Prize in Chemistry 2020 for this CRISPR discovery. As I told, this is a immune system of the bacteria for simple understanding. Okay, so this two enzyme they found is a very important for the drug discovery. One is the Cas9 and another one is Cas12a. The Cas9 particular enzyme we will tell it will act on wherever G present in the nucleotide, the whole genome sequencing. Okay, once again here is the challenge. As I told, the whole genome data will be like a 5 MB to 10 MB, the size of each genome size. Okay, in this we want to find only 20 base pair, which is a good target can be used to clone or to knock out to get the new drug. Okay, here only once again AA play a role. So AA is already he feeded the things which are the target. If you work very well, they develop certain tools. If you give it that your whole genome into the particular tool, it will tell which target will be give more efficient results. So AI driven target can be decided. So which is took a month wise to find out the which is the target. Now it's happening within once again 30 minutes in this place once again. So another drug as I told, enzyme I told nuclease enzyme called Cas12a. This Cas12a is a very new enzyme. It's recently discovered by 2015 only. Okay, from the bacteria called Acidomonaspora and Lachospirae, and 2013 they used Prevotella and Francisella bacteria. Okay, what this speciality of this nuclease enzyme, wherever T is the starting point, it can capable to go and cut. So as I told, the starting one, the Cas9 wherever G is there, it can go and cut, and Cas12a wherever T is the starting point, it will go and cut. These are two unique enzymes currently play a major revolution in drug discovery area. Recently, Caspase, one new enzyme they launched, CRISPR-based therapy for sickle cell anemia. So you all know maybe the term sickle cell anemia is nothing but genetic disorder. See, we have all the other treatment for bacteria, fungal infection, parasite. This is all what everyone heard. But so far, for any genetic disorder, so far we don't have treatment. But after this CRISPR discovery, now recently, 2 months before, okay, the US company launched first CRISPR-based therapy for sickle cell anemia. So that a big achievement has happened. Okay, so as I told, 2013 only this new enzyme is discovered. In very short span of time, it applied in several fields like nucleic acid detection, genome editing, even steganography, okay, DNA assembly and genome regulation. Okay, steganography, I don't know how many people heard previous time to convey the military people to share their information by different way, right? Nowadays, information shared by DNA. Some secret message they will insert in the DNA and it decode by this CRISPR enzyme. Such a level technology is spent. For that, also the AI driven tool one is using to decode this. So then only they can read by the particular military people only to share this information. Okay. So here I want to tell the important current application is CRISPR based diagnostic. So COVID time, all we remember, everyone when afraid, oh, whether I have COVID or not, right? Even the instrument we had and technology we had, it took one day. The one day everyone, you can imagine how much pressure we had in life. Oh, whether I had a COVID or not, tomorrow I will die. Such a level the frustration we went because even though diagnostic method is are there to predict that results, it take huge time because particular expertise needed to predict the results. And even the diagnostic, the qPCR, we will tell biology instrument, it take to run minimum 2 hours to hello.
Yes, sir, you can continue.
I think something is okay. Yeah, some voice in between is coming. No?
Yeah, someone's mic is on. I'll turn it on. Turn it off.
Yeah, yeah. Okay, thank you, ma'am. So the biggest challenge, as I said, that the time consuming and the prediction of the data, right? So what recently the people done, even this is a work done by our team, the method called catfishing we developed. Okay, using this catfishing method, we can clone up to 150 kb. Okay, because this particular enzyme can able to create that 8 sticky end base pairs. With this, we can able to create the big sticky end and able to clone very easily. Okay, for that first we CRISPR enzyme called Cas12a we cloned and we expressed successfully. This is a work first time we done by our team and optimization. As I told, the T should be the starting point, right? So along with the TT, what are the enzymes? Starting point should be there like that. We carried out the then TTA, TTC, this kind of target also it's working like that we found. Okay, and what kind of buffer and how much time it is needed to cut one DNA. There's an interesting point we found that only 5 to 10 minutes enough to cut one DNA. Okay, generally the restriction enzyme maybe you studied in your school time. Once again, the molecular scissor we will tell. Biological people know very well. ECOR1, ND3. Those enzyme and all, it take generally that 1 hour to 4 hours, depends on the enzyme. But here is the case is only 5 to 10 minutes. That's the interesting results we observed the short span of time. Then we created the reaction first to make a trial, 5 to 20 kb small gene cluster we took, and we see the clone effect, and it show very good results. Then why we can't try the silent gene, which I told? Okay, but the problem of that silent gene is generally the size will be big. It's more than 100 kb. Just imagine the gene size will be more than 100 kb. The cloning is very, very tough. Okay, using this CRISPR methodology, first time the 80 kb silent gene is successfully expressed. Okay, the enzyme, the antibiotic called as a surgamide. Surgamide is the drug is used for cancer treatment. Okay, it is 87 kb DNA molecule we successfully express using this method. Okay, this is the sequence verification and the confirmation by gel and others. So, okay, this is worked. Then what about the big size of this next gene clusters? Then we go to the candicidine. Candicidine is the more than 87 kb. So 139 kb, just imagine. That also we got one positive clone. But when we repeated, we are unable to achieve further, but we are optimizing further to go to the more size how it can be work. Okay, so that means what the CRISPR method, okay, along with this modern technology, AI and driven support, we successfully find the new drugs like a surgamide, candicidine. Okay, like that several drugs, silent gene activated, and new drugs are discovered. Okay, along with that, I told that the diagnostic issue with you, right? As a COVID, as I told, it take a huge time to predict the results and running time also. As I told one word to everyone, 5 to 7 minutes only take to clear the target DNA, use the word. Based on that, we developed a method called cat smeller for arthritis people. So we all know that arthritis is the biggest issue. Like it's nothing but old people will always tell I have the knee pains and that, right? Is most of the people have like arthritis related. It's have around 150 types of arthritis are there. Starting stage, we can't predict this. Only we can able to predict immunomodulatory markers. Only the severe condition, when go higher stage, only we can able to predict. The starting stage is very tough to predict. Okay, but using this method, we can able to predict nanomole level target. Remember, what is the unique thing in this? Means nanomole level prediction. That means the disease starting stage itself. In biology term, we will tell acute stage. The starting stage itself, we can able to predict within 10 minutes. Just imagine. For this, no need any expertise, anything. Simply, if we put it within 5 minutes, like we all know that the blood test biosensor, like a blood glucose meter, we all know, put it in blood. If you band, it will show the readings, right? Like that, it will show the readings. So anyone can be predict. So this technology we successfully transferred to the company. They are about to commercialize this product soon in the market. Okay, but what is the sad thing about this? This all the technology mostly with the foreign countries like the US, China only leading this. But currently, our team is working. Recently, Dabadu Chakrabarti, you know that he recently filed, got one patent for F1 Cas9. Okay, so along with that, we are also working to find the new CRISPR enzyme like Cas3 enzyme from Actinobacteria. Our team working how it can be used for Indian public use like genetic disorders and other diagnostic it out. This new enzyme can be used. So we are working very seriously on this project now. So next important thing is the microfluidics. I think if any engineer people is there, you know much better than me. Okay, so generally any reaction volume, if you do 1 ml, 2 ml, any enzyme reactions, you need more volume and very tough to do it. The current term is a lab on chip. It's nothing but if instead of doing like in chemistry lab, we all remember, right? Titration. We put the big biurate and it will drop one by one. 10 ml, 20 ml solution will be there. Slowly we'll open the biurate and it will drop one by one until the color turn will white, right? So it take huge time. For small titration itself take 1-2 hours or 3 hours to our school time chemistry lab experiments, right? But the microchip now no need such a volume. Just 1 or 2 microliter is enough. Okay, and load it in the microchip within 5-10 minutes it can be done. Okay, now the prediction along with the micro fluidics, the AI is attached now, predict the results. So once the reaction happens, the AI will tell is a positive or negative, that's it. So after CRISPR method, the AI based microfluidics biggest boom now. Slowly in India, now only this methodology is booming. Foreign countries already they're going beyond this to technology. The microfluidics also our team is currently working and we're developing CRISPR based microfluidics technology with integration with AI. Currently, we are working on this project. Okay, so we are about to complete soon. So maybe next year we can see in the market our product too, the AI-based microfluidics technology. So how microfluidics working, you can have a look on this, the small video. So this is called droplet-based microfluidics. You can see.
Microfluidics studies the behavior of fluids at the micro to nanometer scale. Microfluidics can miniaturize the work of a whole lab onto a microfluidic chip less than a few inches big. One thing microfluidics is great at is making consistent droplets of fluids. They can be used in cosmetics, chemistry, biology, and even as medicine. To make droplets the regular way, take a beaker filled partly with water and partly with oil, two liquids that can never mix. When you stir, droplets start to form. They are of different sizes. When using this technique at an industrial level, inconsistent droplet size can be an issue, and some of the liquid will be lost because you do not control the quantities precisely. Now, imagine being able to control the exact size and quantity of droplets with a faster and more precise process. Microfluidic equipment can do that. For the same experiment, you need a basic microfluidic chip, a see-through slate with engraved channels only a few nanometers. In the microfluidic channels, you can control how you make the droplets. Put the fluid for droplets in one channel. The other fluid will pinch it from both sides and make tiny drops of fluid that collect in the form of the droplets. Their size is strictly controlled. There is no waste of product, and the process is fast. You can make up to 1,000 droplets per second. Great. But why make droplets at all? Well, this miniature droplet is actually a microreactor. It can be made of any type of fluid, and we can drop what we want to observe inside. This microreactor droplet can be used for many things: cosmetic creams and lotions, supramagnetic nanoparticles, diagnostic tests and assays, mayonnaise and low-fat food with air-filled fat particles, controlled medicine delivery with biodegradable microparticles, and many more.
So I hope you understand that how the microfluidics will make a big revolution in future discoveries. Not only that, as I told, drug discovery, cosmetic, in all the fields play a major role in future. Okay, so the next is the AI in healthcare. As I just now I shown some of the things. Diagnostic imaging is the biggest things. Like if the TB patients, generally they will take CT scan. One of the example I want to give that. Okay, when the CT scan happen, it take one day, then we should be in queue. Next day only the particular doctor will come and it will predict this is a normal chest X-ray or is a TB. To differentiate, it take huge time. Now the AI-driven tools are developed. So when the CT scan happen, within 5 minutes, it will predict that this is a normal one and TB. So then immediately the doctor can enter into the treatment. Okay, such a level revolution is happened in healthcare sector. So current state of AI in healthcare, as I told, precision diagnostic, automation, everything is even DNA isolation too. Everything is going by automation. Okay, for example, biology people know, like a particularly deep sea and volcano sampling, and all, we'll get only pinch of samples. Okay, from that the isolation DNA is very very challenges, very rare sample. Now the AI driven the DNA isolation tools are came. If we keep it small sample, within 1 hour it will isolate DNA and give to us. Okay, and next revolution happening like a generative AI and all. Okay, and framework. Okay, so efficiency, everything can be improved a lot after when AI enter into the healthcare. Okay, and AI solution also day by day is improving a lot. So this is the way the AI play a major role in biotechnology, healthcare. You can see they play a huge role. Okay, and it saves a lot of times and quickly prediction of diagnostic as well as the treatment. It helping a lot to support the doctors as well as the researchers. That is the way AI play a major role. Okay, so coming October 7 to 9 with Bio E3 conference we are conducting both hybrid mode online and offline in Manoranjana University in New Delhi. Here we kept one of the concept is Bio AI, world Indian and worldwide overall working in AI in drug discoveries and others. They are coming in person, as well as we are providing that overall Bio AI related projects you have in your hand. You can submit our Society of Chemical Synthetic Biology providing Symbio grant exclusively for BioAI this year. Okay, so even though last date completed, we extended the date for another 10 days. Overall, have any idea related to BioAI, you can apply your project immediately to our society. So our society mail ID is www.scsbindia.com. Please go through that site and apply your BioAI related ideas and get funding from that. Okay, so this is our entire team. So, and not only that, our Argene company, we can also working CRISPR bioreactor, nanopore related, and all sequence service we are doing. And your students or faculties who are all interested, you can come and get training from our labs, and you can send your students also. This is our mail ID and contact number. And thank you for your listening. So if any queries, I'm happy to answer, guys.
I'm Niveta, sir.
Thank you,
Can I
sir.
ask you?
Yeah. Yes, ma'am.
Sir, you have shown the last slide. You are working in the CRISPR bio AI bioreactor like that. Are you working in any drug related molecules like that, or any actinomycetes based product?
Yes, ma'am. So currently we are working on Actinobacter because, as I told, we have 10,000 Actinobacter whole genome. We have a lot of data with us. Not it's only our lab alone, and along with that, we also have NCB a lot of data, right? So with this all, we are finding new drug molecules. So far, 23 new lead molecules we found through this AI-based tools. Now we are working by experimental proof concept. We are working.
Okay, sir. Sir, are you giving training in CRISPR also, sir?
Yeah,
Faculty?
yeah. For faculties and things, we are giving a training too. If a group of peoples or individually you can approach us,
Okay, sir.
we are giving training for all the sectors.
Sir, how many days the duration will be, sir? CRISPR
And
like that?
depends on that. What are the techniques you are asking? Minimum to learn for only designing alone means 1 day is enough.
Okay, sir.
Complete hands-on experience means 2 days to 5 days, ma'am. Depends
Okay, sir.
on your available. We are giving the things and what kind of techniques can be covered. We are giving details.
We can approach anytime, no problem, sir. Customized like that, or any.
Exactly, it's a customized map. So
Okay, sir.
anytime you can approach. So we are conducting all over the year.
Okay, sir.
Okay, so we are conducting in any time, ma'am.
Okay, sir. Thank you, sir. Thank you.
Thank you. Any other questions?
There is one question in the chat box, sir, by Dr. K. Jayanti.
Agency, okay. Can you find uh, economic order quantity for the buffer and time? Yeah. So that's why I told now that after the microfluidics new technology came now, 2 to 3 microliter only I said now instead of the ml level reaction we can be use only few microliter. Okay, so it saves a lot of buffers as well as the time, as I told, which is happening 24 hours in like a microfluidics kind of technology. Okay, it's completed a very short span of time, 30 minutes. Just imagine, and buffer also the volume is just 2 to 3 microliter because some enzyme, molecular enzyme, and some are very costly, right? Even for basic to conduct our biological experiment, you put ligation also, you put 2 to 3 microliter. Each microliter will be cost if you calculate 4,000 to 5,000 rupees, right? So that can be saved using the microfluidic kind of technology. So it's possible.
Okay. Thank you, sir.
Thank you.
Sir, again, I'm Niveta, sir. One more doubt. Sir,
Yeah.
you told me that AI-based tool for genome sequencing analysis, next-gen sequencing and all, whether
Yes.
we can use or only you people for research purpose, at present it was there, sir.
No, ma'am. Since we are company, we are developed. This is subscription based, ma'am. So
Okay.
now all the colleges are we are giving subscription. We want to pay yearly some subscriptions, like how bioinformatics and other tools we are paying.
Yes,
Like
sir.
that, we are initiated this. This all the tools should reach to every students and faculties. So the subscription can be given to you.
Okay, sir.
So you can directly can access from your sitting place itself, ma'am. So you don't
Okay,
need
sir.
any help of us. That's why this AI tool, NGS assemblies play a very important role, ma'am.
Okay, sir. Okay.
Okay, if you are interested for your college, you can approach us. We can come
Okay,
and
sir.
do directly demo to your students and management. Okay, then students can assemble by themselves this advanced technology for their job and their learnings.
Okay, sir. Sure, sir.
Any other questions from participants? Okay. Since there is no questions, so I would like to thank Dr. Karthik for your wonderful and valuable session. I hope all the audience has gained a lot of insights about the usage of artificial intelligence in healthcare. Once again, thank you, Dr. Karthik, for your excellent session.
Thank you, Dr. Shakti.
Participants, now we are moving to the day 4, session 4, which will be delivered by Dr. Chinnadurai Mani from USA. So now I invite Dr. Rashmi for the guest introduction.
It is my great pleasure to introduce Dr. Chinnadurai Mani, Research Assistant Professor in the Department of Cell Biology and Biochemistry at the Texas Tech University Health Sciences Center, USA. He is an accomplished researcher and academician in the field of cancer biology and human genetics. Dr. Chinnadurai earned his PhD in human genetics from Sri Ramachandra University, Chennai. With a strong academic foundation from India, he pursued his passion for advanced cancer research in the US, where he has made significant contributions in the field of DNA repair mechanisms, cancer chemoresistance, and therapeutic strategies. Over the years, Dr. Chinnadurai has held several research and teaching positions, mentoring students and young researchers, while also developing expertise in cutting edge approaches such as 3D tumor organoid cultures and experimental therapeutics. His research excellence is reflected in his numerous publications in reputed international journals, contributions to cancer drug development patents, and his role as a reviewer and editor for leading journals. His dedication to science has been recognized with several prestigious awards, including the Early Career Investigator Award from the Cancer Prevention and Research Institute of Texas, and the Outstanding Faculty Award for Teaching at TTU HSC, and the Trainee Award from the Texas Society for Clinical Oncology. In addition to his research and teaching achievements, Dr. Mani is an active member of professional bodies such as American Association for Cancer Research and American Society for Biochemistry and Molecular Biology. He has also secured competitive funding to support his innovative work in cancer therapeutics. With such an illustrious academic and research profile, Dr. Mani continues to inspire the scientific community with his vision of translating fundamental research into impactful clinical applications. Please join me in extending a very warm welcome to Dr. Chinnadurai Mani as he addresses us today and shares his valuable insights. We welcome you, sir. So your mic is on mute, I think.
Can you hear me now?
Yes, sir. Yeah.
Yeah, thank you so much for introducing me, and thank you, Dr. Shaktivel, for giving me this opportunity to talk to you guys. Do you want me to go ahead and share the slide?
Yes, sir. Please.
All right, let me know if you guys can see my slide.
Yeah, it's visible, sir.
Okay, so good evening to everyone. It's 8:30 a.m. here, but I believe it should be around 7 or 7:30 in India right now. Thank you for attending my talk. My name is Chinnadurai. I am from Texas Tech University here from Lubbock, Texas. And today we are going to explore about IP in the context of biological sciences. And I will also talk about some key differences between the how IP is handled between India and US. I will also go through research projects. I currently work in a lab where we focus mostly on DNA damage and repair pathways and how it influences chemoresistance. I'll touch base with one of our projects that's currently going on. All right. So you all know that IP is a vital concept for anyone working in research and biotech because it allows us to govern whoever discovers something and how these discoveries are commercialized and the balance between public benefit and the private profit. By the end of this lecture, you all will understand the type of IP that is relevant to biology, what can and cannot be patented, how IP laws differ from India and the US. I'll also go through some real world examples of patents and their impact on biotech, ethical considerations, future trends. And this lecture is mostly a comprehensive one. If you plan to work in research, biotech startups, or pharmaceuticals, I believe you will gain some insights from this. So, why IP matters? IP is crucial because it protects the works of scientists and innovators. Without IP protection, someone could copy your work and profit from it without even giving you credit or compensation, which could discharge investment and innovation. Patents specifically provide a temporary monopoly, allowing inventors to recover their investment. This is particularly important in biology, where research is really costly, and also time consuming. IP also facilitates research technology transfers. For example, when universities patent IP, it also facilitates for to transfer their discoveries and license them to companies. So it allows these discoveries to reach patients as treatments or some commercial products. Uh, for example, consider genetically modified organisms like Bt cotton, which is a pest resistant crop, is patented. And we heard in the last talk about CRISPR, which allows for precision editing of genes, is another example that can be patented and used. Monoclonal antibodies, which are widely used in cancer therapy, are patented to ensure companies that invest millions in development can recoup those costs. In essence, IP incentivizes innovation while creating a framework for commercialization and social benefit. So there are different types of IP. Understanding the historical development of IP in biology helps us grasp why these laws exist today. I believe in the 19th century, patents were primarily used for chemical compounds, dyes, and simple drugs. For example, chemical dyes invented for textile manufacturing were protected by patents, giving inventors exclusive rights to sell and manufacture them. I believe in 1970s, the IPs were more transformative. With the advent of recombinant DNA technology, scientists could combine genetic material from different sources, creating entirely new sequences. This was the foundation of modern biotech. The first patents of recombinant DNA technology allowed universities and companies to protect these innovations. The 1980s and I believe in 1990s saw a boom in companies such as Gentech, Amgen, and Biogen. These companies relied on patents to protect their inventions, attract investors, and bring therapies to market. Today, CRISPR-Cas technology is a modern example. It allows precise editing of genes and has led to a high-profile patent dispute between UC Berkeley and the Broad Institute. If you guys are interested, you can go read about this online, what's the dispute is that's been going on. This also shows how critical IP is in determining who can develop therapies, start companies, and actually commercialize the innovations. So there are four main types of IP that is relevant to biology. The first one is patent. The patents protect inventions such as molecules, genes, diagnostic methods, and production processes. For example, the method to product recombinant insulin is patented. I don't know how GLP-1 is. I don't know if it's popular in India, but it's so popular in US now. Everybody takes, even if they don't have diabetes, just for weight loss. So these new discoveries that are patented, and the companies who are making this are making a lot of money right now. The second one is copyrights. These protect written works, softwares, and databases. For instance, bioinformatics softwares that are used to analyze genome is copyrighted. The third is trademarks, brand names like Nike, Reebok, McD, KFC. These brand names and logos are important for not only these type of companies, but are also for pharmaceutical companies. Humira, which runs genetic analysis in cancer patients, is protected as a brand. The next one we can talk about is trademarks. trade secrets. These are the confidential methods, protocols, or formulation. They are not disclosed publicly, but for example, the exact formulation of a biological manufacturing process may be a trade secret. And the last one is the trade secrets. And each type serves a different purpose, but collectively ensures the innovation is recognized and protected. So, what and all can be patented in biology? Not everything in biology can be patented. To be patentable, an innovation must be novel, non-obvious, and should have some industrial applicability. The compositions of matter, this includes molecule, proteins, or chemical compounds created or modified by scientists. For instance, a newly engineered enzyme with industrial application can be patented. Methods and processes. Techniques or methods like a new way to produce a protein or a diagnostic assay for a disease, those can be patented. Genetically modified organisms like Bt cotton, pest resistant cotton, or engineered bacteria, they also can be patented if they meet novelty and usefulness requirements. Diagnostic methods, a new method to detect a biomarker for any type of cancer or any kind of disease, is actually patentable. However, there is a difference. The naturally occurring genes or proteins that the human did not modify, which occurs naturally, they are generally not patentable unless it's modified or applied innovatively. So I'll just take you through the patent application process, how it is obtained. The process actually starts with an idea, of course, followed by lab research to develop and validate the inventions. Uh, a patent search is then performed to ensure the invention is novel. This is crucial because if a similar invention is already exists, then the patent will be automatically rejected. The next step is filing the patent with the appropriate patent office, either nationally or internationally. The patent office examines the application for novelty, non-obviousness, and industrial applicability, and they may request clarifications or amendments. Once it's granted, the patent holder has exclusive rights for a set of period, which is usually around 20 years in US. I'm not sure about what's the date set in India, but the maintenance fees are required to keep the patent active. Enforcement is the responsibility of the patent holder. They must monitor and, if necessary, take legal action against infringers. This process is really expensive, time consuming, and it requires careful planning, but it is also essential to secure a legal protection for innovation. I believe in US, we have to spend somewhere around $2,000 to $5,000 to do a patent search. We usually outsource, I think. So, Indian IP laws. Let's go through some Indian IP laws in biology because they have some unique features. The Patents Acts of 1970, amended in 2005 to comply with TRIPS, allow patents for novel inventions that have industrial applicability. Natural life forms such as unmodified plants or genes cannot be patented. Only microorganisms that are modified or engineered can be patented. An important distinction is that discovering a gene or a protein in nature is not at all sufficient for a patent. The invention must actually involve modification or application that adds any value. India also emphasizes in public health. Compulsory licensing is allowed for essential medicines to ensure accessibility and evergreening of patents, where companies try to extend patent life without substantial innovation. It's actually restricted in India. Now let's go through some US IP laws, especially the IP laws that are relevant to biology. The US has one of the most robust and the historically permissive kind of IP framework for biological innovations. The US patent law, the legal foundation behind the US patent law is that it allows patents for any new and useful processes, machine, manufacture, or any matters This explicitly includes genetically engineered organisms, recombinant DNA sequence, and biologicals. This legal framework incentivizes innovation by granting inventors exclusively the right, typically for 20 years from the date of filing. Naturally occurring genes. This is a little bit of in the gray side because historically the US allowed patents even for isolated natural genes. India doesn't allow that, but US allowed patents for isolated natural genes. For instance, BRCA1 and BRCA2 genes. Whoever works in the field of biology or cancer must have heard about these genes. These genes are highly mutated in both breast, ovarian, and cancer, and also they also found that it is highly mutated now in pancreatic cancer as well. So these genes were actually patented by Myriad Genetics. This meant that Myriad had exclusive rights for these mutations in these genes, which led to high costs for patients and limiting testing options for them. However, the Association for Molecular Pathology, they took it to the Supreme Court in US, and the court ruled this happened in 2013, and the court ruled that the naturally occurring DNA sequences, even when isolated, cannot be patented because they are products of nature. But you can still patent a complementary DNA, which is synthetically created and does not exist naturally. This distinction clarified what constitutes a patentable invention in genetics, at least in US. Next, we will move on to the biotech innovations incentive. The US law, it strongly encourages private investment in biotech. Companies and universities can secure patents for genetically modified organisms, recombinant proteins, novel production processes, editing technologies like CRISPR. These protections allow a company to attract investors, fund expensive research, and commercialize discoveries. For example, Genentech, they are the ones who invented the recombinant human insulin and growth hormone, and the patents protect these inventions, enabling the company to establish a biotech industry in the US. I mean, they didn't discover the human insulin, but they got the patents. CRISPR technology, the patent dispute between UC Berkeley and the Broad Institute highlights how crucial IP is for determining commercial and research insights. And there are some biologicals and monoclonal antibodies. Patents cover not just the product, but also production methods. For example, monoclonal antibodies, which is very famous in cancer therapy, PD-1 inhibitors, PD-L1 inhibitors, all those things. So the key takeaway from this slide is that the US patent system is designed to maximize innovation and commercialization. It provides broad protection, allowing biotech companies to invest heavily in research and development. Unlike India, where public health considerations can limit patent scope, the US system primarily prioritizes innovation incentives, sometimes at the cost of accessibility. As we move forward, you will see how this contrasts sharply with Indian IP laws, which are more restrictive on natural genes and more flexible for public health needs. All right, now let's compare the key differences in terms of IP for biology between India and USA. Let's start with the natural gene patents. In India, you cannot patent naturally occurring genes. Only genetically modified organisms or engineered microorganisms are eligible. This ensures that discoveries in nature remains accessible and cannot be monopolized. However, in US, as we discussed before, naturally occurring genes were initially patentable until the case between the National Molecular Association versus the Myriad Genetics Company in Supreme Court in 2013, where they ruled that only synthetic constants like cDNA are patentable. This difference highlights India's more restrictive approach, but also it focuses mostly on its public accessibility. In India, if something is discovered, the Indian government wants the public to have more access to it. But in US, it's they give more priority to the discoverers rather than the publics. Next, we will see what's the difference between India and USA in terms of genetically modified organisms. Actually, both the countries allow patents on GMOs, but the process is stricter in India. For example, BT cotton, it was patented and commercialized in India after proving novelty and industrial applicability. In the USA, the GMO are broad, covering the organism itself, the method of protection, and even certain uses, allowing biotech companies extensive protection. Next, we'll go through the biologicals or recombinant proteins. In India, the biologicals and recombinant proteins are patentable if they meet novelty and industrial application criteria. In USA, patents covers not just the product, but also the production processes and methods. For instance, genetic recombinant insulin and growth hormone patents are protected for both the molecule as well as how they produce those. We will go through public health flexibility. India includes provisions to ensure access to medicines. That is very good because we are developing this for public. So I personally believe this should go to the public. Compulsory licensing can allow other manufacturers to produce a patented drug in case of public health is needed. This ensures affordability and availability, particularly for life-saving drugs. However, in US government prioritizes innovation incentives. While there are mechanisms for access, the system generally favors patent holders, sometimes resulting in higher drug prices, but encouraging investment in new biotech startups. I have known some people from here in Texas. It borders with Mexico. They just travel to Mexico to get their insulin or their GLP-1 injections because it's way cheap in Mexico than in US. All right, we will finish the slide by going through the evergreening of patents. So evergreening is when minor modification to an existing patented drug are used to extend its patent life. India has strict restrictions to prevent this, ensuring generics can enter the market sooner. In USA, however, the incremental innovations can be patented, which can effectively extend market exclusivity and incentivize continuous implementations. So the key takeaway from this is that these differences reflect the contrasting priorities of two countries. India emphasizes more on public health access, whereas US emphasize mostly on innovation, commercialization, and investment incentives. If you develop a biotech invention where you file the patent and how you structure your claims can significantly influence your commercialization, collaborations, and accessibility. I think it's important for whoever is planning to start a startup or whoever is planning to do some kind of IP IP protection. It is important for you guys to understand these differences. So now that we have discussed real life examples of IP in both in US and India, let's synthesize some information and draw lessons for some of the faculties or the researchers here. So the scope of patented material, the US allows a broader scope of patenting. Not only you can patent genetically modified organisms, but also synthetic constructs such as cDNA, production methods, and applications. In India, on the other hand, is more restrictive. As I said before, the natural genes cannot be patented, but engineered organisms can be patented. In US, let's see how the legal disputes are handled between the US and India. In US, for example, the CRISPR-Cas patent battle show that the legal strategy is a core part of the research commercialization. You may need to navigate disputes, licensing agreements, and collaborative IP rights. But in India, the focus is mostly based on the public benefit. Compulsory licensing ensures essential medicines remain affordable and evergreening restrictions prevent undue monopolies. Next, we will talk about the international agreements related to IP. Understanding these agreements is essential because science and innovations are increasingly global now. Many discoveries have potential applications beyond national borders, and international agreements help harmonize IP protection worldwide. TRIPS, trade related aspects of IP rights, is an agreement and the WTO, World Trade Organization. It was implemented in 1995. It sets a minimal standard of IP protection that all WTO members countries must follow. TRIPS covers patents, copyrights, trademarks, designs, trade secrets, and geographical indications. For patents, TRIP mandates that invention in all fields of technology, including pharmaceutical, should be patentable if they are novel, innovative, and they're industrially applicable. The second one is the Patent Cooperation Treaty. This is another international agreement that's been established. This is administered by World Intellectual Property Organization that simplifies filing patents in multiple countries. Filing a PCT application allows you to submit a single international patent application that can enter national phases in over 150 members countries. So this gives inventors more time, usually 30 to 31 months from the initial filing, to decide in which countries to seek actual protection. The PCT system is particularly useful for academic researchers and startups who may not have the resources to file separately in multiple countries immediately. India and TRIPS. India is a WTO member and is obliged to comply with TRIPS, but it implements some aspects more restrictively, particularly in pharmaceuticals and biotechnology. For example, as I said before, India does not allow patents on natural substances or traditional knowledge, even if they are isolated or extracted, whereas the US and Europe may allow patenting of isolated DNA molecules or synthetic derivatives. As we discussed before, India also emphasizes public health and access to medicine, allowing the companies or allowing the mechanisms like compulsory licensing, which TRIP permits actually under certain conditions. For the faculties and students here, this means that an innovation patentable in the US may not automatically be patentable in India. Conversely, innovation aimed at public health or accessibility may have strategic advances in India's restrictive framework. If you are planning a patent in biotech, pharmaceuticals, or any globally relevant research area, you need to understand both the TRIPS framework as well as how your Indian laws implement it. Filing a PCT application can give you the flexibility to protect your inventions internationally without the immediate high cost of multiple national filings. Understanding these agreements is just not the legal knowledge, but it also, I would think, it's a strategy for deciding where, when, and how to patent your research. So now let's explore the specific challenges that arise when dealing with IP in the field of biology. Biological research represent unique hurdles compared to other fields like software or mechanical engineering because of its complexity, cost, and ethical implications involved. So anyone who works on cancer research or any research in biology, they will already know how cost consuming is research to do in biology. Patenting biological inventions, especially in biotech, pharmaceutical, or genetic engineering, can be really expensive. The cost include patent filing fees, legal fees, and translational fees if you are filing for international patents, and maintenance fees over the lifetime of the patent. For example, filing a single patent in the US can cost anywhere from $10,000 to $20,000, and maintaining it over 20 years adds additional costs. Filing in multiple countries can increase these costs exponentially. The second thing is the complex inventions are hard to patent. Uh, Biological inventions are often complex and can involve multiple components such as genes, proteins, pathways, or organisms. Determining what is patentable requires understanding novelty, inventive step, and industrial applicability, which can be difficult for highly sophisticated or overlapping inventions. Ethical concerns, for example, genetically modified organisms, uh, patenting a gene. Biologically, this raises unique ethical challenges that can impact patenting. For example, patenting human genes, genetically modified organisms, or stem cell technologies can often face ethical debates and legal scrutiny. In some countries like India, natural genes and traditional knowledge cannot be patented uh, because they reflect concerns about monopolizing naturally occurring substances, which is perfectly understandable. Um, the last thing we will discuss is balancing knowledge sharing versus protection. Academic research encourages publication and dissemination of knowledge, but early publication can jeopardize patentability if the invention is disclosed before filing. This happened to one of our researchers who actually used Gardasil vaccine for treating cervical cancer patients. He published it as soon as he got some enough data from the patients, but he came to know later that he cannot patent this. He cannot patent this procedure because he already published it. So the practical advice that you can take from this slide is that work closely with your institution's technology transfer office to identify which discoveries are worth patenting and which can be freely published, because you have to come to a decision whether this patenting this particular project which you're going to patent is going to be really helpful or if you can make money out of it. Keep thorough dated lab notebooks and experimental logs to provide legal evidence of invention, and understand the ethical and legal boundaries of your research field, especially regarding genes, stem cells, and GMOs. Uh, Budget for patenting cost early if you plan to commercialize your work or seek international protections. Yes, the next we will go through the ethical considerations in IP, which are particularly significant in the field of biology, especially when we are handling with naturally occurring genes or proteins. Unlike pure technical fields, biology often deals with life forms, natural resources, and human health, which brings a set of moral responsibility alongside legal consideration. Gene patenting is one of the most debated ethical issue in biological IP. And can a company or individual claim ownership over a naturally occurring a gene? I don't know. Many argue that genes are part of humanity and it shouldn't be owned. I also believe that nobody should own a gene. On the other hand, isolating and characterizing a gene, creating a synthetic version, or developing a therapeutic application may justify a patent protection. The policies differ by country. As we discussed before, US genes are patentable until the recent court rulings, which has actually restricted this. However, in India, it explicitly disallows patents on natural genes. So ultimately, the ethical IP involves balancing private rights with public interest. Innovation should not only reward inventors, but also contribute to society progress, public health, and sustainable development. So I can just talk about the ethical thing, but I think you guys can think of each case. You can go through and think about it, whether they are ethically relevant for you or not. Next, to make the discussion of IP more concrete, I will go through some real life example from modern biology, how the patents are disputed or how it is helping. I'll start with the patent dispute surrounding CRISPR-Cas9 editing technology. I believe our last speaker spoke about, he gave some introduction about CRISPR, and I think we had a session about CRISPR yesterday or day before yesterday. But CRISPR-Cas is a revolutionary gene editing tool that allows scientists to precisely modify DNA in living organism. It has enormous potential in medicine, agriculture, and biotech, enabling therapies for genetic diseases, developing for genetic disease-resistant crops, and many more things. There is a patent dispute actually uh, between UC Berkeley and the Broad Institute. I'll just give you a brief background about this. When CRISPR was first developed, two major institutions filed patents. The one is UC Berkeley, the other is the Broad Institute at MIT Harvard. This dispute was centered around who invented the technology first and who had the rights to specific applications in eukaryotic cells like human and plant cells. Initially, the Broad Institute received the patent for eukaryotic applications due to faster filing with expedited review, while Berkeley had earlier claims in prokaryotic systems like bacteria. Actually, this conflict have escalated over the years, involving patent offices in the Europe, US, and as well as internationally. So the importance of licensing, it's a basic thing. You should license whenever you get some good research products. But once the patents were established, licensing become critical. Licensing determines who can commercialize the technology, whether in startups, pharmaceutical therapies, or agricultural products. Companies like Editas Medicine, Intellia, and Caribou Biosciences negotiated licensing from Broad or Berkeley, enabling them to develop CRISPR-based therapies. Without clear IP rights and licensing agreements, startup would risk legal risk disputes and financial losses. Uh, can you give me just a second? My daughter is crying. She doesn't want to go to school. I'll just go convince her and come back.
Yes, sir.
Yeah, just 2 minutes. I'm sorry.
Yes, yes, yes.
I'm so sorry. She cries every day when she goes to school. So, all right. Okay, let's see about this CRISPR-Cas dispute. So this case actually illustrates how IP directly affects both the research and commercialization. Researchers must navigate patent landscapes carefully to avoid infringing on the existing IPs. So the key takeaway from this slide is CRISPR-Cas licensing disputes show how IP has real-world consequences on science, business, and public health. Licensing is essential for startups and commercialization of biological discoveries. Early awareness of IP helps researchers plan projects that are both scientifically innovative and legally secure. So next, we will talk about another real-life example, Genentech and the recombinant insulin. Genentech was one of the first biotech company to commercialize a recombinant protein by using genetically engineered bacteria to produce human insulin. They actually revolutionized diabetes treatment. Their patent covers both the insulin and also the method of producing it, giving Genentech exclusive right for several years. This allowed this company to attract investments and scale production, and they're one of the topmost biotech companies here now because they had a very strong IP protection incentives when they were a startup company. mRNA vaccines, I think in India you all must have received some protein vaccines or attenuated vaccines, but here most of the people in US received mRNA vaccines. Moderna or the Pfizer, they developed COVID-19 mRNA vaccines, and the patent covered the modified RNA sequences, the lipid particles that will deliver the RNA inside the cell, and the method of delivery. This IP framework incentivizes companies to invest in high risk cutting edge research and also enable the rapid commercialization. I think we went through this before, so I will just skip this slide. So let's see a real life example in India. So far we have seen the real life example in US where the companies or the people who bought the license, they have more control over the products. But in India, it's a totally different thing. The Indian government wants the public to have more access in relative to the companies or whoever bought the patents. So the real life example of IP in India, let's talk about BT Cotton. Um, so BT Cotton, it was initially provided by Mayco Monsanto Biotech. BT cotton is basically a genetically modified cotton variety that produces a bacterial toxin to protect against bollworm. In India, the patent was granted based on genetically modified organism, not the natural cotton gene. The introduction of BT cotton actually revolutionized agriculture, increasing yielding and reducing pesticide use, while the patent encouraged the company to invest in research. The Ayurvedic and herbal formulations, those are traditional knowledge protection. So India has a rich knowledge of Ayurvedic medicine. To prevent misappropriation, India has implemented traditional digital knowledge libraries. This ensures that the ancient knowledge cannot be patented elsewhere. For example, turmeric's wound healing properties and neem extracts are documented to prevent foreign patents on traditional remedies. This demonstrates a unique aspect of Indian IP, protecting public domain and cultural knowledge while encouraging innovation. Affordable HIV and AIDS drugs, CIPLA, and compulsory licensing. India has been a global leader in producing affordable genetic drugs. CIPLA famously manufactured low-cost antiretroviral drugs, making HIV treatment accessible worldwide. India's IP laws allows for compulsory licensing, as we discussed before, in case of a public health needs. So this ensures life-saving drugs are not priced out of reach. This is one of the major difference in the IP laws between India and US. So Indian companies like Bharat Biotech or Serum Institute of India developed recombinant hepatitis B vaccines and played a critical role in COVID-19 vaccine productions. And these patents were filed for novel recombinant methods and delivery systems, not the natural viruses themselves. So the key takeaway from this slide is that the Indian IP encourages innovation but limits monopolies on natural products and traditional knowledge. The law strikes a balance between commercial incentives and public health needs, making drugs and biologicals more affordable and widely available. So, in summary, the US IP laws prioritize innovation and investment, and the Indian laws carefully balance innovation with societal needs and accessibility. Now, let's see a real world case study from India that shows how IP biotech can come into conflict with the public interest. So I'll go through briefly about this BT content before. I went through briefly about BT content before. So BT content is basically a short for Bacillus thuringiensis cotton. This is a genetically modified variety of cotton that includes a gene from a soil bacterium. The gene allowed the plant to produce its own insecticide, especially effective against bollworms, which are a major threat to cotton cobs. In the late 90s and 2000, Monsanto, a US-based biotech company, actually developed and patented BT cotton seeds. They introduced this into India through a joint venture called Mako Monsanto Biotech. The IP protection applied to the gene itself, the transformation method, and the use of technology in cotton. I believe this was a major success in India. The farmers saw that their pesticides uses has dropped and their yields were actually increased. By 2014, over 90% of the Indian cotton fields were planted with Bt cotton. However, the things got complicated on the IP and legal front. India, being a TRIPS compliant country under WTO rules, does respect patent rights, but it also has provisions to ensure public access to essential technologies, especially in the field of agriculture and as well as in medicine. Over time, dispute arose over royalties and licensing fees. The company who introduced this to India charged technology fees per pack of seeds, which many Indian states considered too high. In 2016, the Indian government stepped in and it slashed the royalty rates by law, arguing for farmers' welfare. Monsanto pushed back, claiming this violated the patent rights, but this case went all the way to Indian Supreme Court, which ultimately ruled that the plants and seeds, being natural products, are not patentable under Indian law, even if genetically modified. So this is a kind of a big difference in how IPs are approached in US law, whereas versus in Indian law, where in US this would have been the company would have got the be on the winning side, but in India it's usually the people or the public is on the winning side. So this highlights for all the scientists and students here today the importance of understanding not how just to patent, but where and under what a framework you should patent those. Uh, BT cotton still remains India's only GM crop approved for cultivation, and it continues to stir debate among IP, biosafety, and farmers' right. So next let's discuss about important distinction in IP, uh, patenting biologicals versus small molecules. This is a critical concept in pharmaceuticals and biotech industries. Small molecules, they are chemically synthesized compound with low molecular weight. The examples are aspirin, paracetamol, antibiotics, all those. Once the patent expires, generic manufacturers can replicate the molecule relatively easy, which is why these small molecule drugs often face quick generic competition, because the small molecules are simple. The focus is mostly on molecule itself, not necessarily on the production process. Next is biologicals. The biologicals are rare, complex molecules derived from living organisms. The great examples would be monoclonal antibodies, enzymes, hormones, vaccines, and many other things. Biologicals are strategically important because competitors cannot simply copy the product even after the patent expiry without developing their own complex manufacturing processes. Next, we will see the manufacturing processes. For biologicals, the process is as important as the product, because if you find something and if you don't know how to reproduce it, or if you don't know how to make it again, then that's going to complicate your whole research. This ensures quality, safety, and exclusivity. For example, monoclonal antibody therapies like Herceptin relied heavily on the way how they produce these antibodies, giving the company extended market exclusivity beyond the basic molecule patent. So in modern medicine, biologicals are becoming more prominent, highlighting the growing importance of biotech IP knowledge for students and early career scientists. So the key takeaway from the slide is that the small molecules, they are simple, molecular focused patents, easy generic competition, whereas the biologicals that are complex, that has complex processes, or highly exclusivity or manufacturing, those process innovation are critical or crucial in biological IP strategy. And understanding these differences helps guide research, commercialization, and patent strategy. So now let's discuss an important concept that every biotech entrepreneur should understand: the patent life and market exclusivity. This determines how long a company can profit from its innovation before competitors are allowed to enter the market. In most countries, including US and India, patents are granted for a 20-year period starting from the date of filing. This applies to both small molecule and biologicals. The clock actually starts ticking from the filing date. You have to remember that it's not from the date of approval or the date of commercialization. It starts whenever you file for an application. While a patent may last for 20 years on paper, the effective commercial life of a biological or a drug is usually shorter. Why? Because regulatory approvals take time. For a small molecule, clinical trials and FDA review can take around 10 to 12 years. So you lose almost more than half of your patenting lifetime in just getting approved from FDA, at least in US. As a result, by the time the drug reaches the market, a significant portion of the patent life may already be consumed. The US has an additional protection beyond the basic patent, market exclusivity, particularly for biological and orphan drugs. For biologicals, you have a 12 years of exclusivity for the original biological under the BPCIA Act. And the drugs also can be, I think you can extend it for 7 years independent of the patent, meaning that even if a patent expires, the company may still prevent competitors from entering the market for over a 7 years period. So the companies must strategically plan patent filings to maximize the effectiveness and commercialize and increase the commercial life of their products. Delay in filing or clinical development or regulatory approvals can actually erode potential revenue. So the key takeaway from this slide is that 20-year patents are standard, but effective market life is shorter due to clinical and regulatory delays. And the US biologicals enjoy additional exclusivity periods that extend protection beyond the patents. Understanding patent and exclusivity strategy is actually essential for innovation, commercialization, and career planning in biotechnology and pharma. So next we will go through the IP rights over medical devices, which are pretty much similar to the other things, whatever we have discussed so far. These devices often combine hardware, software, or something biologicals or sensors, making IP protection complex, but they are important component of the IP. So integrated medical devices are systems that combine physical hardware, software, and the example include if you have a wearable glucose monitor that measures blood sugar level, and they transmit the data to a iPhone or a smartphone. That can be patented as well. Robotic surgical systems that combine mechanical arms or software control and imaging, and lab on a chip devices for rapid diagnostic testing, those are all can be patented. So the protection of these design of the device, for example, the structure of a robotic arm or the functionality like the algorithms that they use to detect anomalies in a patient, or a novel integration of components like combining microfluids with the embedded software. So to summarize, the integrated medical devices required multi-phased IP protection due to all these components that we discussed now. A careful IP strategy ensures innovation, safety, and commercialization potential. And students and researchers need to think strategically about patenting, licensing, and possibly an international protection if you are using anything that is will be helpful for the patient. And international protection would actually maximize the impact. Now let's focus on the challenges and opportunities in IP for young researchers. I think we are almost at the end of my talk before I go through some of my research slides. So one of the complex challenges is understanding how IP laws and the patent works. IP laws are often technical and it varies by jurisdiction. For a student or a young researcher, knowing what is patentable, how to write claims, and what constitutes novelty or innovation can be overwhelming. For example, in India, the natural genes or traditional knowledge cannot be patented, whereas in US, a synthetic DNA and engineering genes are patentable. Misunderstanding these rules can lead to missed opportunities or rejected patent applications. Limited funding, filing and maintaining patents can be really expensive. We spent around $30,000 to $40,000 whenever we submit something to patent in our university. For young researchers in universities or small startups, the cost of international patents and legal fees and the maintenance fees can be really a significant barrier. This is why many researchers must rely on the technology transfer offices or seek institutional support for patenting. Risk of IP disputes and legal costs. Patent litigation can be extremely expensive, especially in high value fields like biotech and pharmaceuticals. So the key takeaway from this whole cell, because I don't want to go through each and every point in this, the key takeaway from this slide is that while IP management can seem daunting, it is an essential skill for researchers who wants to maximize their impact of their work. The scientists who invest time in learning IP law, filing patents, and strategizing collaboration will have a competitive advantage. For students who are interested in global research, understanding the difference between jurisdictions, balancing publications versus patenting, and leveraging IP for social benefit are critical skills to learn. I'll skip this one and we'll to the conclusion. So as we discussed, the IP is very crucial for protecting biological innovation. On a whole, India looks more restrictive, but because it wants public to enjoy all the benefits that they get from the IPs. But US is morely permissive, which is lenient and more, and it moves more towards the startups or the inventors, and it encourages commercialization. And understanding IP laws guides research, collaboration, and biotech entrepreneurship. And we went through the real life examples of how CRISPR, Gentech, and mRNA vaccines in US and the BT cotton in India. All right. So my IP part of the talk is over now. I will move on to my research project. So the title of the project is Prexasertib, which is actually a check1 inhibitor. It synergizes with a PARP inhibitor called olaparib, and this works mostly in triple negative breast cancer. This study was done in triple negative breast cancers. So people who worked in biology, it will be really relevant for their insights, but I don't know, people who are non-biology, it might be a little hard for you to understand, but I will try to simplify the whole presentation. So we all know that the cancer has become a global epidemic and the leading cause of death worldwide. It is actually more deadly than AIDS, TB, and malaria combined. And in 2019 alone, it is estimated that around 1.7 million new cases will be diagnosed, mostly as a prostate, lung, and colorectal cancer in males, whereas breast, lung, and colorectal cancer in females. So these are the currently available treatment options for cancer therapy. Surgery remains as the most effective treatment with minimal side effect, but there are places where you can't do surgery. If someone has a leukemia, then surgery is not an option. Or if somebody has a tumor in their brain where it's hard to reach, then surgery is not an option. So radiation and 90% of the chemotherapy works by inducing DNA damage in cancer cells, which eventually leads to cell death. Targeted therapy can also be used, like EGFR inhibitors in lung cancers. Some hormones like male sex hormone testosterone have the ability to drive tumor growth in prostate cancer. And the recent hot topic, immune therapy, which includes vaccines and PD inhibitors or PDL1 inhibitors, are also available for cancer treatment. While targeted therapy, hormone therapy, and immune therapy remains as an optional treatment, invariably, almost all cancer patients undergo either radiation or chemotherapy at some point as an adjuvant or a neoadjuvant therapy. Radiation or chemo is they are actually very successful in shrinking the tumor and increasing the patient's life expectancy. However, on the long run, they do not completely cure the diseases, and most of the time, this results in tumor recurrence. You must have seen someone who's been under a chemo or a radiation. The doctor might have said that the cancer is completely gone, but in 5 to 10 years, in most of the cases, you would see the cancer has come back again, unfortunately. And chemo resistance has been identified as the most important reason for disease recurrence. There are multiple pathways a cancer cell can become chemo resistant. Drug effects, for example, there are numerous studies show that the ABC transporters, they're really active in the cancer cells and has the ability to reduce drug activity by either reduced uptake or increased drug effects. Drugs can also modify the chemotherapeutic agents. For example, glucose S transferases are overexpressed in many cancers, and they can actually modify cisplatin, which is a platin based drug that's given for gynecological cancers, by adding a thiol group onto the cisplatin and decreases its affinity towards DNA and inactivates it. Cancer stem cells these are a subpopulation of cancer cells characterized as cells with tumor forming ability. One of the hallmark of cancer stem cell is that it can remain dormant without replicating in the tumor microenvironment. Since most of the chemo or radiation kills only actively dividing cells, cancer stem cells can actually escape from these drugs and be a cause of relapse later. EMT transition also makes the cancer cells to gain migratory and invasive properties and also becomes more resistant. The remaining two are the increased DNA repair and the cell cycle arrest. These are the main focus of our lab, so I'll just go into detail about those two. So it has been estimated that around 1000 to 1 million molecular lesions happen in a single cell in a given day in all of our body. I don't want you all want to get panicked about it, because almost all of this will be repaired immediately. But yeah, at any given time point, there are these many molecular lesions in a single cell in your body. How this is happening, there are various sources have been identified. For example, the normal DNA replication errors, reactive oxygen species, UV, chemo or radiation, all those things could do this. I don't want to go into detail, but I just want you guys to understand that out of all the damages, the double strand break is considered the most lethal, because it has been proved scientifically that for a cell to die, it has to have at least 1,000 single strand breaks, something like this. But even if you have 4 to 8 double strand breaks, the cells would die. And because it is important for also maintaining genomic stability, since it is so important, the double strand breaks are actually repaired by two different pathways. The one is non-homologous end joining, the other one is homologous recombination. Non-NHEJ is very simple. It happens mostly during G0, G1 phase, where there is no complementary DNA. It removes the damaged DNA and just simply attaches them. This is often an error-prone repair pathway with always small deletions, but the cells are not dividing at that point, so that's the best thing that they could do. Unlike non-homologous end joining, homologous recombination is highly regulated, which happens mostly in cells during or after replication. Since it is highly complex, it is being regulated by various proteins. First, the double strand breaks are recognized by ATM or ATR kinases, which recruits the other proteins. These are kinases that just go phosphorylate the other DNA repair proteins like BRCA1, BRCA2, RAD51, and they all assemble at the point of DNA damage, and they will just repair the damages. So the scientists and researchers who worked on this DNA repair mechanisms understood the importance of creating a double strand break. So they came up with a target called PARP protein. The PARP is an active member of single strand break repair mechanism. Inhibition or trapping PARP to DNA results in failure of recruiting repair proteins at the site of single strand breaks. The unrepaired single strand breaks get converted into double strand break during replication, and this has to be repaired by homologous recombination mechanism. Even though the PARP inhibitors were initially approved in patients with BRCA deficiency, later it was found that deficiency in any other HR proteins like ATM, ATR, RAD51, or FANGD2, they all showed synergistic or synthetic lethality with PARP inhibitors. So far, there are 4 PARP inhibitors that have been approved by FDA. Olaparib to treat breast, ovarian, and prostate cancer, talazoparib to treat breast cancer, and rucaparib to treat ovarian cancer, and niraparib also to treat ovarian cancer. I'll skip this slide because it will too much for you guys to understand. So I'll just give a glimpse of how we started this current project. I was working on a project before, and we were evaluating a drug called CF10, which was synthesized by one of our collaborator, Dr. William from Wake Forest Medical College. What we observed is whenever we treated with this CHEK1 inhibitor, we found the level of RAD51, which is an important component in homologous recombination, it's going down. Whenever we see this RAD51 going down, we also saw increase in gamma H2AX. Gamma H2AX is nothing but a fancy name for phosphorylated H2AX, which just tells you how much double strand breaks are there in that cell or in that particular cell line. So we did most of these experiments in breast cancer cells because breast cancer is the most common type of cancer in women, and the FDA has approved PARP inhibitors in breast cancers first. So we thought this would be a right model for us to study. So the first thing we did is we wanted to reproduce our observation in the other experiments. So we did a concentration dependent treatment of prexasertib, which is basically a CHEK1 inhibitor. And whenever we increased the dose, we treated them for 24 hours. We found that the RAD51 level and the BRCA1 levels were going down. However, the gamma H2AX or phosphorylated H2AX levels were increasing, shows that the DNA repairability of the cancer cell is going down, and the cancer cells now have more DNA damage. We also did a time frame study. We used 250 nanomolar prexasertib, and we treated for 0, 6, 12, 24, and 48 hours. We found by 24 hours, both the BRCA and RAD51 levels are less and gamma HTX is more. So one of the critical things that we have to check while we are evaluating HR protein is the cell cycle profile. Normally the HR proteins, they are expressed mostly during S and G2M phase because that's where it has a template DNA ready to carry out the homologous recombination. So some people might argue that the prexasertib is arresting the cells at G0, G1 phase, and that's why you are seeing the RAD51 levels are going down. So to rule out that possibility, we did a cell cycle analysis, and we found that whenever we treat with prexasertib, we see the cells are mostly arrested in the S phase of the cell cycle. So this rules out the cell cycle mediated RAD51 downregulation. We did MNSA. I actually learned MNSA when I was in India doing my PhD. This was one of the most important assay, and it has been very well known that micronucleus assay, the CHEK1 inhibitors induce MN. We just confirmed in our hand whether it's the same or not. So when we treated with prexasertib, we saw small micronuclei. These are actually the damaged part of the nuclei that are not anymore attached to the parent nuclei. This is DMSO, just control cells, and when we treat with prexasertib, we saw increased micronuclei. We wanted to study what's the mechanism behind downregulation of homologous recombination, how exactly prexa is inhibiting RAD51. So we treated the cells with MG132, which is a proteasomal degradation inhibitor. So what we observed is whenever we treat with prexa, we saw the RAD51 levels going down, or BRCA1 level going down. But when we inhibit the proteasomal degradation with MG132, we see those levels coming up. We were able to reproduce in multiple cell lines, which confirm that the protein stability is basically affected by CHEK1 inhibitor. We used a different proteasomal inhibitor. This also showed the same result. We did RT-PCR. It did not show much change in the mRNA expression. It showed some difference in few cell lines, but not all cell lines showed a significant change in the mRNA expression. So overall, this gives an idea that prexasertib affects RAD51 stability post-translationally, not during its translational activity. Now we know prexa downregulates RAD51. We wanted to be sure whether it actually has any effects on homologous recombination. So this setup is actually a patented setup. We usually pay $60 to $100 to buy these plasmids from a company here and use this. It was actually developed by Dr. Maria Jesin in NIH, I believe. So this, what we do is, this is called a DRGFP HR assay. So first we transfected the cells with the DRGFP plasmid and we selected them using puromycin. This plasmid has 2 GFP, but both are non-functional. The first one is not functional because it has a ICE1 sequence inserted in it. And the second one is non-functional because both the N terminal and the C terminal is completely cut off. So both of them cannot express any GFP. After puro selection, we transfect the cells with ISE1. ISE1 is endonuclease. It's not expressed in human gene. This is not present in human gene. It's produced only by bacteria. However, this ISE1 removes the ISE part from GFP1, inducing a double-strand break. Now, this GFP1 has to do a homologous recombination to get that particular part of the gene from GFP2. And now the gene is actually complete now. So it can actually express GFP. So to make it easy, if you have homologous recombination, you will see more GFP positive cells. If you don't, then you will not see more GFP positive cells. And this is our result where we gated to remove all the autophosphorylated cells. And this is DMSO treated cells where you see a lot of GFP positive cells. But when we treat with prexasertib, we see that there is less GFP positive cells, almost 50% reduction in the GFP positive cells. This confirms our RAD51 downregulation actually affects the homologous recombination efficiency. So next our question is, let's combine this with PARP inhibitors, because if homologous recombination is not working, then PARP inhibition should sensitize the cancer cells when we combine them both. So that's what happened here. So this one is just olaparib, prexasertib. You see increased DNA damage. Next we used olaparib. It also shows increased DNA damage. But when we combine both of them, we see more level of damages as measured by gamma H2AX in 3 different cell lines. So my boss, he likes to confirm same thing with different methods. So we used Western blot initially to confirm the protein expression. He also wanted me to use immunofluorescence to confirm the foci formation is also going down. So this is TMSO. Prexa, you don't see any foci. With olaparib, you see BRCA1 foci. But when you combine prexa and vola, there is no BRCA1 formation. Same with RAD51. However, you see increased gamma H2AX. This slide shows that BRCA1, RAD51, both DNA repair proteins are going down when you combine Prexa with Vola. However, the gamma HTX, which is a marker for double-strand break, is increasing. Finally, we had to use Comet assay. Technically, the fragmented or the damaged DNA will not be able to bind to the nuclear DNA, and we all know DNA is a negatively charged nucleic acid, and it moves towards we just put it in an electrode and it moves towards the positively charged. And when it moves, the damaged DNA with a small size will run faster than the whole nucleus, which will form a comet-like structure. That's in the solar system, so that's why it's named as comet assay. So as you can see, these are DMSO treated cells. There are not much DNA damage. However, in Prexa, you see some little DNA damage. Vola, you see some little DNA damage. But when you combine both Prexa and Vola, you see a lot of DNA damages. We used colony assay to confirm our results. I'll just directly go through the data. So this is just olaparib treated cells, increased concentration on the x-axis and survival fraction on the y-axis. But whenever we add prexasertib, we see the combination kills more cancer cells compared to the individual drug treatment. This is just a combination index which shows that whether the combination is synergetic or not. Actually, this is done by a software where you have to enter the survival fraction, and that will give based on some formula whether the CI is less than 1 or more than 1. So if this combination index value is less than 1, that means they are really synergistic. If it's more than 1, they are mostly additive, or I think they're mostly additive. But we see some of the values here are really synergistic in MDA MB231 cells. In 453, most of our combination showed a synergistic interaction between CHEK1 inhibitor and PARP inhibitor. So finally, to summarize, PARP inhibitor induces DNA damage, mostly single-strand breaks, which then converted into double-strand breaks whenever the cells replicate. The prexasertib, which is a CHEK1 inhibitor, inhibits both the cell cycle arrest and also it inhibits homologous recombination by downregulating or affecting the protein stability of RAD51 and BRCA1. So this ultimately leads to cell death. So to conclude, since olaparib is already FDA approved and the prexasertib drug is already in phase 2 clinical trial, we believe that this combination will be translated easily. We also found some racial disparity where African Americans have in US have more RAD51, and incidentally they also have more challenging tumors, breast cancers compared to the other population. So currently we are using mouse models to establish our study, whatever we found in vitro, and to see if they are reproducible in vivo. And we also want to evaluate RADFID. We also getting some patient sample from our collaborators, so that will help us to tell whether RAD51 expression, what we found in online databases are real or not. So with that, I will just stop my slide sharing, and I'll be happy to answer if you guys have any questions.
Thank you, sir, for the very nice presentation and an insightful talk on different aspects of IPR, and also for sharing your research work with us. Sir, I have one question. Could you please explain how the royalty is distributed in US?
Oh yeah, so royalty in US, it's actually it's very different between the universities. So what happens in our university is, for example, if I find a drug and if I sell it to a company, and if that company gives $100 for my product, I will get 40% of that $100. The 30% goes to the Office of Research Commercialization, those are the office from our university who actually piles the patents and they take care of everything. So 30% goes to them, and 10% goes to my department. I'm from cell biology and biochemistry department, so 10% goes to them, and 10% goes to the school. So the Department of Cell Biology and Biochemistry comes under School of Medicine. So 10% goes to the school, and the remaining 10% goes to the university.
Thank you, sir. Is there any other question from the participant side? Or if you have any questions in the research aspects also, you can ask, sir. Okay, so if there are no questions, we'll wind up the session, and there's a feedback link that is posted in the chat box, and you can all fill it, and we'll meet tomorrow at 6 p.m. exactly. Thank you so much. Thank you, sir.
Thank you so much.
மட்டும் இருக்கு அது மட்டும் கேள்வி பண்ணா அவ்வளவு முடிஞ்சு இன்னொன்னு